Okay, everybody, we're going to be looking at chapter 17, the special senses. In chapter 16, we talked about the general sensations and how they are incorporated into the brain. And the special senses are incorporated into the brain in a similar way, but they're special because their receptors are going to be very unique and they have more complex pathways of getting the information to the brain and being interpreted. So we're going to take a look at each of these with our senses. Now, before we get started, I do want to kind of do a comparison of general senses versus special senses. Now, so guys, when we look at special senses, receptors for special senses are for like smell, taste, vision, hearing, and equilibrium. So if we think about our five senses, this is all of our senses except for touch. Touch are considered general sensations. However, another sensation that's here that we don't normally talk about as being one of our senses is equilibrium, which we are going to hit on today. Now, these are anatomically distinct, so each of the receptors are going to look very different. Unlike what we saw with the general senses, where each of the neurons kind of look similar, the difference was whether they're free nerve endings or encapsulated. These are all going to look very, very different, and all of them are concentrated in the head region. Touch is throughout your whole body, pain is throughout your whole body, itch is throughout your whole body. However, when we look at the special senses, they're going to be located in the head region. I mean, you have your vision, you have your smell, you have your taste, you have your hearing, and your equilibrium is also part of your ear. All right, so that's part of in the head as well. Now, these are specific afferent pathways, meaning they're sending the information to the brain to different translation sites in the brain. So we're going to see that there's going to be a primary center for your hearing, a primary center for your vision, and so on. So when we look at general versus special, general senses include somatic sensations like tactile, which is touch, thermal, temperature, pain, and proprioception. These are also going to include your visceral sensations, the ones that are associated with your internal organs that a lot of times are subconscious. You're not really aware of them. They are scattered throughout your entire body, and they have a relatively simple structure. On the other hand, your special senses include smell, taste, vision, hearing, and equilibrium. They're concentrated in the head. We just talked about that. They are anatomically distinct, which means they also are functionally distinct, and they are more complex, which makes them special. So what we're going to do is we're going to focus in this chapter on these. Now, olfaction and gustation or taste, we're not going to spend a whole lot of time on because these are the two simplest, and we're going to spend more of our time on vision and hearing. But we do want to talk about these two first. Now, olfactory is smell, and we've talked about with the cranial nerve. This is going to go with cranial nerve number 1, the olfactory nerve. You also have gustatory, which is taste. The impulses from both of these are chemical senses. So these are chemicals that have to dissolve that we can then detect. Okay, chemicals that are in the air or chemicals that are going to dissolve in our saliva for our taste buds for us to taste. They travel not only to the cerebral cortex, so not only to this top part of the brain, the cerebral cortex, but they're also going to be associated with the limbic system. And if you'll recall from chapter 14, the limbic system is that system that deals with memory and emotions. So a lot of times this is why when you may smell something or taste something, it might trigger a memory or it might trigger an emotional response, especially if it's like you've lost somebody recently and maybe they had a certain kind of food that they made and that reminded you of them. It might trigger some sort of emotions that come up with that. Gustation and olfaction work together, but olfaction, guys, is much stronger and more sensitive than your taste. So smell is a lot more sensitive, but this is also why it's important that they go together. If you've ever had like a cold or a stuffy nose where you can't smell, do things taste a little different? They don't taste the same. They may not even hardly taste like anything. This is because smell and taste go together, and smell is the more important side of it when we talk about it being sensitive and stronger in how it sends its sensations. So let's talk about smell first, or olfaction. So the first thing I want to talk to you about is the anatomy of the receptor for the olfactory, for smell. These receptors are bipolar neurons. Now, when we look at bipolar, bi means 2. So this is where you're going to have one end that's going to collect the information. Then it's going to have a axon kind of structure to its body, and then its main axon to talk to the next area. This type of neuron was discussed in chapter 12 in your book. Now, these particular neurons are located in the nasal epithelium in the superior portion of of your nasal cavity. So they're way up high, kind of like past the bridge where my glasses are sitting. So they're way up high up in this area, and so a smell has to get up in there. This is why when your nose is kind of clogged, it has a hard time getting up there and you have a hard time smelling things. These are considered a first order neuron. You'll recall back from chapter 16, we talked about in sensations, there's a first order neuron, a second order neuron, and a third order neuron. These are the first order neurons. So this is going to be the area that's going to pick up that sensation due to the environment changing. On these nerves, guys, there are some special structures called olfactory hairs. These olfactory hairs are actually cilia or extensions of the plasma membrane, which increases their surface area so that they can actually come in contact with more chemicals. Now, because of this, their threshold is very small. It only takes a few molecules to be dissolved in touching these hairs in order for us to smell something. Okay, now, of course, the more chemicals that come in, the stronger the smell might be. So this might be if somebody walks by you that just put on a bunch of perfume or cologne and a lot of those chemicals come in, you're going to smell it a lot more than if it was just a little bit. But we still can distinguish different smells only by a little bit of the molecules being present. However, guys, with smell, adaptation does occur very quickly. We get used to the smell very fast. You lose sensitivity. An example of this is like if you've ever run over a skunk, or you've gone past a place where a skunk has recently been run over. The smell is there and it like fills your whole car. You can totally tell it's a skunk smell. But eventually you get used to it. This doesn't mean the smell is gone. Okay, because let's just say you stop and you pick somebody else up and they get in and they're like, did you run over a skunk? And they can smell it. You can't because you're used to it. Your cells have adapted. All right, and so that's what kind of happens. That's why sometimes if you have like a Scentsy or something on that has a smell in the room, you may not smell it eventually, but then somebody else comes in and they can smell it very strongly. Okay, so we lose those adaptations very quickly. Now, guys, with olfaction, we can actually interpret tons of smells. Okay, I mean thousands of different types of smells. This is why some people who have a really good sense of smell can sit there and they can like smell something and they say, oh, that has cinnamon in it, and that has this or that has that. This also helps people who can taste those different kind of herbs or spices. The smell helps them be able to do that, where they can distinguish little differences in them, whereas some of us may not have that ability. My sense of smell is pretty keen, and it has been since I was pregnant with my first daughter. With my first daughter, if I smell even just a little bit of peanut butter, it would make my stomach turn. I could not handle it. And so I had to tell my students a lot of times, please don't eat peanut butter, or if you do, please wash your mouth out before you come to class, because I could smell it even if it was a tiny little bit of amount. Okay, because we have the ability to distinguish between lots and lots of smells. Now, these olfactory receptors don't do their job on their own. Okay, remember neurons a lot of times have cells that help them. Well, these guys have supporting cells that are found in the epithelial tissue of the mucous membrane in your nose, and they are going to provide physical support to these receptors. When I say physical support, they're going to actually help keep them in place physically, help hold them in place, but they're also going to offer them nourishment. They're going to help give them glucose, oxygen, that kind of thing. They're going to kind of take care of them. And they are going to also electrically insulate them so that they can send the signal to the proper place as we see how the signal travels from the receptor to the olfactory bulb. In this area as well, there's also some basal stem cells. Now, you may have heard, recall back in the skin chapter that there was that stratum basal, that base layer in the skin that were like the stem cells, and they keep replacing the skin after you slough it off and lose it. Well, these basal stem cells here are going to provide you with new olfactory receptors. Your olfactory receptors only live for about a month, and then they need to be replaced. Now, this is very unique because in most cases with our nerves don't get replaced. However, with olfactory senses, we actually do see them replaced every month for the most part. In this area too, you're going to see olfactory glands. Now, these are important because they're located in the connective tissue and they're called Bowman's glands. These are going to actually produce mucus. Now, why do they need to produce mucus? Why do we need mucus on these cells? Well, guys, the mucus is actually going to go across the surface and it's going to trap the chemicals. When they trap the chemicals, it dissolves them and lets them come in contact with these olfactory hairs so that we can actually detect the sensation, detect the odor. So it's really important there so that it moistens the mucous membrane, but also allows for the odorants, the smells, to dissolve so that we can actually send the signal to the brain. Now, if you take a look here, you can see that olfaction is shown here in the picture. It shows you how the tissues look in the first one over here with the connective tissue, the basal cells, the olfactory glands. They're all labeled here for you. I'm not going to expect you to memorize this, but if you notice, you can see that it is a type of epithelial tissue with some connective tissue. Down here at the base, you can see the olfactory hairs. Now, where is this tissue located? If you look at the picture that's on the right-hand side, you can see that it's at the very, very top of your nasal cavity, way up high. But you'll also notice that it comes in contact very closely to the brain. Okay, this is important because it's going to be sending the signal to the olfactory bulb and the olfactory tract. If you'll remember, when you dissect the brain, you saw those as they came down. Okay, so you have the tracts that are there, and you can actually see them coming across, and that's where they're going to detect these signals. Now, this picture is found on page 574 in your book. All right, so how does olfaction work? Okay, so how do we look at the whole idea of how do we send this signal? We saw the anatomy side, now let's look at the physiology side. How does it work? When an odorant molecule, when that chemical actually dissolves into the mucus, it stimulates an olfactory receptor. When it stimulates that receptor, it creates a, kind of generates a potential. Now remember, we have to see that these general potentials have to be added together until we finally reach that threshold where we send the signal. So there has to be enough chemicals present in order for you to detect it to send the action potential down the nerve. Once that action potential goes down the nerve, the axons of several olfactory receptors merge together and they kind of bundle together with connective tissue. They pass through the cribriform plate. Now, if you'll recall, the cribriform plate is part of the ethmoid bone. If you'll remember, the ethmoid bone kind of looked like a spaceship in a sense. It had the crista galli, which stuck up, and then it had this plate that came across here. This is the cribriform plate. You'll also recall that there were what we call olfactory foramen. Olfactory foramen were the small little tiny holes, and that's where these nerves are going to pass through. Each bundles of these axons that come together are called the one olfactory nerve. You're going to have one on the right side and one on the left side. And there's about 40 olfactory nerves bundled together to create this cranial nerve 1. Because remember, olfactory nerves, cranial nerve 1. Now, the axon terminals of the olfactory nerves are going to extend through those tiny holes in the cribriform plate that are called the olfactory foramina, and they are going to come in contact in synapsis, a connection, with the olfactory bulb. So once the signal is sent, they're going to send the signal to the olfactory bulb, and that olfactory bulb is going to send it down to the next area. Olfactory bulbs, they lie immediately superior to the cribriform plate, just right above the cribriform plate. One's going to be on the right side of the crista galli, the other is going to be on the left, and these olfactory bulbs are made out of gray matter. So recall, gray matter is going to be the cell bodies. Okay, these cell bodies are going to connect to the axons, which are going to be the tracts. These are going to talk to the olfactory tracts. The olfactory tracts extend posteriorly towards the back, okay, going back away, okay, towards the back of your head. They're going to carry these impulses to the cerebral cortex as well as the limbic system. Now remember, the limbic system is for emotion and memories. Okay, and this is why with smell, sometimes it can trigger those memories. Now, on this slide, guys, I wanted to show you kind of what the last slide said, but more in a flow chart. This one talks to this one, which talks to this one, and so on. So we have the olfactory nerves. You can see them here. They're going to be picking up the signal by the olfactory hairs, and they're going to then collect it and send it to the olfactory bulbs, which you can see located just right above the cribriform plate over here in the picture on your left. Once they take that signal, they're going to send it down the olfactory tract. And if you look in the picture on the right, you're going to see that the olfactory tract then sends the signal to the primary olfactory area. This olfactory area is located in the temporal lobe of the cortex. Remember, the temporal lobes are over here to the side, and you can see that that's where it is heading. Okay, and so this is the sequence of how we get the message from your nose when you smell something to your brain to process what you're actually smelling. Okay, so this is what we're looking at for olfaction. All right, and again, these pictures are going to be found on page 574 in your book. Now, that's pretty much all we're going to look at for smell or olfactory. So now let's switch gears and talk a little bit about taste. Now, gustation, or also known as taste, it's going to be simpler than olfaction. Okay, when I say simpler, this is because we don't actually taste thousands of different types of things. Tastes are going to fall into kind of 5 main categories. Okay, does it taste sour, sweet, bitter, salty, or does it have what we call umami? And you may not have heard of umami before, but umami, guys, is that savory taste, kind of like when you have a juicy steak. If you like steak or something like that, that savory taste, that's what we're talking about with umami. So those are the ones we're going to be looking at here for taste. Now, you have approximately 10,000 taste buds on your tongue. Okay, so a lot of your taste buds are on your tongue. However, that's not the only place they're found. They're also found on your soft palate, okay, towards the back of your throat, your pharynx, which is your throat, and your larynx down here. Okay, you have taste buds that run down almost connecting to the esophagus. All right. Now, one thing to note though is that these taste buds, the number starts to decrease with age. Now, they're very heightened in working in babies. This is why if you've ever tasted baby food, you're like, oh, that tastes so bland. But to a baby, it doesn't. It's actually very sweet or very sour. Okay, this is why sometimes you can get that real extreme reaction when you give a young child a lemon to eat. If you've ever watched their face, okay, where they make that really strong face because of that lemon, that's because they have a lot more taste buds than we do as we grow older. All right, part of that's because of damage to taste buds. We burn them off, like when we can't wait to eat that pizza and it burns your mouth and you can't taste anything for a couple of days. We've damaged some of those taste buds through the years. Okay, but when we look at this, they do decrease with age. Now, each taste bud, guys, is composed of about 50 gustatory receptor cells. Now, you can actually see them in this picture over here. They are called taste buds. They look like a little bud or flower. Okay, so there's a bunch of them all bundled together in this bud type structure, about 50 of them. Now, they're not just going to be these receptor cells. We're also going to see that there are going to be supporting cells located here that help cause that oval ball shape that's there. These supporting cells are going to help them give their structure. But again, just like we saw in the olfactory, they're going to help nourish them, provide them with glucose, oxygen, that sort of thing. Now, there's also some basal cells here, like stem cells that we saw in the nose, and they can become supporting cells around the bud. Okay, these stem cells become those supporting cells. Now, this picture of the taste bud is found on page 577 in your book. Now, guys, each gustatory receptor cell has one gustatory hair that sticks up, all right? And so you can kind of see it here in the picture, and I'm actually going to go back to the slide before real quick, and you can see it here. See how you have the gustatory hairs that kind of stick up? Now, guys, you're thinking, oh, wait, but my tongue doesn't feel hairy. No, they're still just an extension of the cilia, but your tongue does feel rough. That whole idea of it feeling rough is part of this structure that we're going to be looking at with these taste buds. Now, these are microvillus. They extend out through the taste pore to the surface of the tongue, the soft palate, the pharynx, the epiglottis, all those areas we talked about that have taste buds besides just your tongue. Now, each gustatory receptor cell does have a lifespan of about 10 days. So we do see a replacement of them. However, their replacement gets slower and slower the older we get. And this is why we lose the amount of taste buds as we get older. But they last about 10 days. So this is why, guys, sometimes when you burn your mouth with something, like you couldn't wait on the hot chocolate or the coffee or the pizza or whatever, and it burns them, they can be replaced, but for a few days you don't taste very well. Then you start to get your taste back. Okay, so when we're looking at taste buds, guys, there are several different types of taste buds, and they are put together in a structure that we call papillae. There's what we call the valate papillae. There's about 12 of these. They're the large ones. They form in a row at the back of your tongue, and each one of these houses about 100 to 300 taste buds. Okay, so they're very large with lots of taste buds located in this structure. The next ones are called the fungiform papillae. The reason that they get this name is that they're mushroom shaped. That's where the fungi comes from. And these are scattered all over the entire surface of the tongue. And they're smaller and contain only about 5 taste buds in each of them. Then you have what we call the folate papillae. These are located in small trenches on the lateral margins of your tongue. So towards the side, there's these little trenches that these guys are going to be found in. But most of their taste buds will degenerate in early childhood. So early on in childhood, you'll start to lose these. And so again, remember we said that they would decrease. Your number of taste buds would decrease with age, and this is one of the places that that occurs. So in addition, the entire surface of the tongue also has some papillae that are called filiform papillae. And these contain tactile receptors but no taste buds. And so they don't have the receptors for taste, but they do have the receptors for touch. And the whole point of this is to increase friction between your tongue and your food so that as you're chewing, it moves and churns the food to create that bolus, that food into a ball to get you ready to swallow it. And this is why your tongue feels really rough. Now, substances to be tasted must be in a solution because remember these chemicals have to be dissolved for you to taste them. This is why you need the saliva. Not only do we need the saliva to help with digestion, like we're going to look at in the digestive chapter, we also need it there to dissolve the substances, the chemicals, so that we can taste them. Now, there's a threshold that will vary with each different taste or chemical, but adaptation does occur pretty quickly. We lose our sensitivity to that taste, and that's why a lot of times when you're doing like a taste test on stuff, whether it be like cheese, wine, food, or whatever, they'll want you to clear your palate in between, and they'll do this with something that will help like kind of take all those chemicals away to allow you then to be able to taste the next thing that you put in your mouth. So it's really important to do that if you're using them in that sense because they do adapt very quickly. Now remember, our primary tastes are sour, salty, bitter, sweet, and umami, and they are found on different areas of the tongue. All right, so I'm going to kind of show you here. So the tip of the tongue is where you're going to see most of your sweet. Now there's sweet throughout your whole tongue, but this is where they're concentrated, is at the tip. That's going to be the sweet. Just above that on the sides, you'll see the salty. Above that is the sour, and then here at the back of the tongue is the bitter. And so when we look at this, this is where you're going to find it. Now umami is more in the middle of the tongue. All right, so where you see those concentrations. So this is what we're looking at with your different tastes on your tongue. Now, when a gustatory hair is stimulated by a chemical, it's been dissolved in the saliva, it goes to that hair and we actually can detect it. This causes a release of a neurotransmitter from the bottom end of the gustatory receptor cell. So it's going to send a neurotransmitter to the next cell to tell it what it has tasted. This neurotransmitter then causes a nerve impulse to travel along a neural pathway to cranial nerves. Now, there are 3 cranial nerves that it can talk to. The first is cranial nerve 7, which is the facial nerve, and you can see it located here. It's going to be more of the front of your tongue. We also see that there's cranial nerve 9, which is the glossopharyngeal. This is going to be kind of towards the back, kind of middle area. And then you also see cranial nerve 10, the vagus, which is the very back part of the tongue. Okay, and so or actually it's not even part of the tongue, it's more towards the back on your pharynx area. So these are going to conduct the signal. So now we're going to see that it's not just one nerve that can pick this up, there's going to be 3 different options. Now, with these 3 cranial nerves, they are going to send their impulses through the medulla, okay, because we saw that these come off the medulla. They're going to travel through the thalamus, which you can see here, which is the darker portion. They're then going to travel through the limbic system and the cerebral cortex to the primary gustatory area. This is the area where we're going to see taste. It is at the base of the somatosensory cortex of the parietal lobe, okay, and now this is important because this is also the area that deals more with the texture of the tongue, the tactile receptors of the tongue. So it's going to be in close contact with that area in the brain. All right, so this is going to finish up that kind of pathway for the gustatory. So we've talked about smell, we've talked about taste. Now we're going to spend the majority of our time on vision and hearing. So let's talk about vision next. So when we're talking about vision, our visual perception depends on a number of things. The first is your eye itself, okay, the eye structure and the eye itself. There's also the accessory structures of the eye, like your eyelids, the lacrimal apparatus that makes tears, your eyelashes, all those types of things. But then it also depends on the optic tracts. That's going to be the nerves that carry the information to your brain. And then, of course, the primary visual cortex in your brain for interpretation, so that it can interpret what you're actually seeing. Okay, so there's a lot of different things that come into play here with your vision. Now, photoreceptors are the special receptors that are located in your eye, and they can actually catch what we call photons. Photons are how light travels. Now, they travel with what we call the electromagnetic spectrum, or EM spectrum, or what we call radiation. Now, the light that we actually use to see can be measured between 400 and 700 nanometer wavelengths, and you can see it here in the diagram. It's a very small portion, but this visible light, guys, can be broken down into the colors of the rainbow. rainbow. If you'll look here, this is like what we would see with the colors of the rainbow that we call Roy G. Biv, right? Red, orange, yellow, green, blue, indigo, violet. These are those colors. That's why when we take light and we pass it through a prism or through water, it can create a rainbow. Now, the electromagnetic spectrum does not just have visible light on it, okay? It has what we call gamma rays. Gamma rays have the highest energy and they can cause damage to our cells. They don't turn you into the Hulk like we see gamma rays do in those movies and the comic books. It actually causes damage to your cells and can cause cancer. We then see one with a little less power is X-rays. X-rays, though, can still be dangerous. Now, the couple of X-rays you may get a year or something like that on injuries is not a big deal. However, you may notice when you get X-rays, they'll put that lead vest on other areas that aren't being x-rayed. That's to protect your cells from damage. But guys, if you're going to be like an x-ray technician or a radiologist, you're going to be doing x-rays all day long. And so it's very important that you protect yourself from this high energy wave. And that's why a lot of times they go into a room, and in that room it's lined with lead to protect them. Then you have UV rays. And guys, you've heard about UV rays. These are ultraviolet rays. These are the rays that come from the sun that actually can sunburn you, and they can cause cancer as well because they're high energy. But guys, if you remember, ultraviolet means above violet. So the next area when we look at the visible light, you see violet, then indigo, blue, green, yellow, orange, and red. After red, guys, you have infrared. That's below red on the spectrum. Guys, infrared is lower energy, and you'll also see that we use infrared goggles to be able to see this. This is where you can see the colors and things like that if you have those types of goggles. Then you have microwaves. Guys, microwaves are really not that dangerous. You might have been told as a kid, don't stand in front of the microwave because it can cause problems and things like that. You would have to be exposed to microwaves for a very, very, very long time constantly before they would cause damage to your cells, all right? Because they're pretty low energy waves. They also then see radio waves. Radio waves are lower energy, but they have longer wavelengths. This is why radio waves can travel over very long distances. You might be in the middle of nowhere and you can pick up maybe at least one radio station. Maybe on the AM side and it's talk radio, but you can still pick it up because these guys can travel through long distances. All right, so this is what we talk about with the electromagnetic spectrum, and we're going to be focusing in on this tiny little section here for visible light. Now, before we get into kind of how the eye works, we need to look at the anatomy of the eye. And we're going to start first with the accessory structures, the structures that help the eye stay healthy. And the first is the eyebrow, okay? So you have your eyebrows. This has a protective function. It keeps falling objects out of your eye, actually. So like something falls kind of here, it can catch it and help keep it out of your eye. The sun, believe it or not, it does help, okay? It may not help a lot. That's why a lot of times we may do this to kind of help keep the sun out of our eye, but it does help because it's on that ridge of our skull. And also sweat out of your eyes. This is why a lot of times when you sweat, the sweat will come off to the side. It'll run around the side and not get into your eye, okay? Now again, this isn't 100%. But guys, eyebrows also make you more aesthetically pleasing, okay? You can Google like celebrities without eyebrows where they've taken the eyebrows off, and you look a lot different without your eyebrows. But not only do they make you look more pleasing, they also are there for a purpose, okay? They are there to help keep your eye healthy. The next one are your eyelids. Your eyelids are also known as the palpebrae. These are protective as well, like your eyebrows. And again, they're to help keep things out of your eye, but keep your eye moist so that when you blink, it helps move the fluid across the eye. Eyelids are they spread your lacrimal fluid, which we call tears, all over your eyeball. And they have an area that's called the tarsal plate. The tarsal plate is a set of connective tissue that supports each upper and lower eyelid. Okay, so with like ladies, you may know this because it's that connective tissue that's right on the edge. This is where we put like eyeliner on. Okay, and maybe even some guys, if you've worn eyeliner before, it's going to go on that area that's a little harder and thicker. That's the connective tissue of the eyelid. Now, there's also on these eyelids what we call the meibomian glands. These are sebaceous glands inside each tarsal plate, and they're there to help with lubrication, okay, right against where the eyelid hits the eye. And guys, one thing to note is your upper eyelid is more movable than your lower eyelid, so that when you blink, your upper eyelid is what comes down. Our lower eyelid does not come up. That would be kind of weird. So what we see here is the upper goes down. And the reason it's more movable is it's attached to more muscles. Okay, so more muscles are attached to it, which allows it to be more movable. The next thing we want to look at is the eyelashes. Eyelashes are also protective like your brows and your lids. They have sebaceous glands at the base of each hair follicle of each of your eyelashes. And these are called the glands of Zeiss, and they help keep the hair follicle and the hair healthy. Now, a problem though is if these glands do get infected, it creates what we call a stye. Okay, so a stye is an infected sebaceous gland that is associated with your eyelashes. Also with accessory organs, we see that there's intrinsic eye muscles. These eye muscles are going to help move your eye. There are 6 muscles that move each eye, and you can see a lot of them here in the picture in the diagram. So there's 6 muscles for each eye that help move your eye. It's in different directions, also help move the eyelid and so on. Now, there's also a structure called the conjunctiva. The conjunctiva is a thin mucous membrane that lies the inner aspect of the eyelids itself. It's continuous with the front surface of the eyeball except for over the cornea. The cornea is the very middle part that allows light to kind of come through. Now, you may have heard a little bit about the conjunctiva when we talk about conjunctivitis, which I'll talk a little more about in a minute. Now, with the conjunctiva, it's composed of stratified columnar epithelium. Now, you don't see this in very many places. So if you'll recall way back from tissues, stratified means many layers. Columnar means they're columns, so they're tall anyways, and there's a couple of layers of them. They're not found in very many places. In these areas, too, you have many goblet cells, which produce mucus. These are going to be the ones that also produce what we call those eye boogers a lot of times that we have in our eyes, or sleep that collects in the corners when we wake up in the morning. Bloodshot eyes are due to an irritation or infection of the conjunctiva. So if they look bloodshot or pink or red, that's what's infected or is been hurt is the conjunctiva, and we call this conjunctivitis. This is due to the vasodilation of the blood vessels. Now guys, conjunctivitis can be viral, bacterial, or it could be due to an irritation like an allergy or like damage to the eye. All right, and so that doesn't have to be contagious, and a lot of times we call this pink eye, but not all pink eye is contagious. All right, if it's bacterial or viral, it can be, but if it's due to like allergies, it's not going to be contagious. All right, and so one of the last accessory structures we want to talk about is the lacrimal apparatus. The lacrimal apparatus is going to create lacrimal fluid, which are what we call our tears. Now guys, your tears are mostly made of water, but they also have a little bit of mucus in them. They have something called lysozyme, which is an enzyme. This enzyme is super important in helping protect against infections from bacteria. And there's also salts. And we know that there's salts because if you've ever accidentally, I mean, I'm not saying you drink your tears on purpose, but if you've actually accidentally gotten a tear in your mouth, you can tell that it's salty. You make approximately 1 milliliter of tears per day. The purpose of these tears is to clean your eye, to lubricate your eye, moisten your eyeball, and protect again against infections, especially bacteria. Now, you have 2 lacrimal glands. They are almond shaped under the lateral portion of your eyelid. So I'm going to take my glasses off just a second and kind of point to where that is. So they're located just right in here, okay? So there are going to be 2 almond shaped structures located just right in this structure. One's a little bigger than the other. Now, each of these is going to secrete its tears via lacrimal ducts. Now, there's 6 to 12 of these in each eye. I don't know why there's the range, but there are 6 to 12. They are going to release your tears onto the upper conjunctiva, which then drains into 2 lacrimal puncta. The puncta, guys, okay, I'm going to take my glasses off again. The puncta are right here in that we would call the tear ducts, okay, right here in the corner. They're the tiny little holes that you can see, and you have one on the top and one on the bottom to the corner of your eye, okay, so they're right there in the corner of the eye. This punctica is going to drain the fluid into what we call the lacrimal canal, which is going to then take it towards the nose. They're going to then drain into what we call the lacrimal or the nasolacrimal sacs, which are located here, and then ultimately into the nasolacrimal duct, which is going to drain the tears into your nasal cavity. This is why when you produce a lot of tears or you cry and stuff, a lot of times your nose will run because that's where a lot of them are going. Also, then eventually your nose will start to stop up because the mucus starts to increase. So guys, when we talk about watery eyes occur when lacrimal fluid builds up. This could be a number of reasons. There could be a blocked nasolacrimal duct, like in the nose, which is due to inflammation, like when you have a cold. So your eyes will get to where they're watery and we'll see them drain out the side instead of draining into the nose. There could be an overproduction of tears. This could be in response to a parasympathetic stimulation, like when you're emotional and you cry, okay, when you're sad. Tears will spill over the edges of the eyelids and come down the side, but they will also continue to drain in the nasal cavity, and this causes them to have causes you to have nasal stuffiness when you've cried a lot. Okay, so that's what we're looking at here, because guys, if we look at this, every hole in your face is ultimately connected. All right, these holes here are connected to your nose is connected to your mouth. In your ears, we're going to see that there's tubes that are connected to your mouth and throat as well, and so everything is kind of connected in these areas, and the eye again is not any different. All right, so now what I want to do is I want to look at the actual anatomy of the eye. With the anatomy of the eye, there's 3 layers to the wall of your eyeball, and we're going to start with the outermost layer first and work our way in. So the first outermost layer is called the fibrous tunic. It is avascular. When we put a in front of a word, it means without or not, and vascular means blood vessels. So this layer actually has no blood vessels to it. This area contains the cornea, which is in the very front of the eye. This is non-keratinized stratified squamous epithelia plus some collagen, okay? And when we say non-keratinized, keratinized is the protein in your skin. It doesn't have that. We also see that it's connected to the sclera. The sclera is the white part of your eye. It covers everything except the cornea. Okay, so the whites of your eyes are the sclera. It's made of dense connective tissue and collagen, and the whole purpose of this layer is to be protective. It helps maintain your eyeball's shape. Okay, so we want your eyeball to be an 8-ball shape. This helps do that. Now, at the junction of the sclera and the cornea, there's a small opening here that is called the sclera venous sinus. Okay, so there is a very small opening between the cornea and where the sclera attaches. All right, and you can see that the sclera is the outermost portion of the eyeball here. This is called the fibrous tunic. The second layer is called the vascular tunic. This is the middle layer, and it's also sometimes called the uvea. This layer has a section called the choroid. It's a dark brown colored structure due to the fact that it has melanocytes in it. You may recall back from the skin chapter that melanocytes are there to help give you pigment to your skin. They have the same structure here. They make this area darker. This lines the sclera in the back of the eye. They absorb light rays. Darker colors absorb more light. Okay, this is why, like when you go outside and you wear a black shirt, it absorbs the light and you get hotter faster than if you wear a white shirt. So this layer there is to help absorb the light as it enters your eye, so it doesn't just bounce around. It actually absorbs it so that we can use it. Okay, so it's not scattered inside the eyeball. This also, because it is the vascular tunic, it means that it has lots of blood vessels, and these blood vessels are going to carry nutrients to the retina, which is going to be the next layer we're going to look at. This layer also has some structures that we call the ciliary bodies. These line the sclerocorneal junction, okay, where the sclera and the cornea come in contact. They have what we call ciliary processes. These are folds in the epithelial tissue. They're very vascular. They have lots of little capillaries for blood supply. They secrete a watery fluid that's called aqueous humor. Guys, aqueous humor is very similar to cerebral spinal fluid. Remember we talked about the coracoid plexus, which releases the cerebral spinal fluid? Well, here we have what we call the ciliary processes, which are going to release aqueous humor into this part of the eye. The ciliary process also hold the iris in place. The iris is going to be the colored part of your eye. It helps hold it in place. Ciliary muscles are also located here. They're circular smooth muscles, and they're there to help focus the lens. So if you look here, the lens is suspended by these little muscles, and they're going to help pull and move the lens to help you focus on what you're looking at. Now, the iris I already hit on is the colored portion of your eye. It's between your cornea and your lens. It has some circular and radial smooth muscle fibers, and they are going to surround the area called the pupil. The pupil is the hole in the center of the iris which allows the light to pass through. Now your iris, the colored part of your eye, is going to regulate the amount of light. It's going to help determine how big your pupil is. Is it constricted and small when there's bright light, or do we need the pupil to dilate when there's low light. The iris is going to allow this to take place. Now guys, this is an autonomic control. You do not have to be consciously aware of this happening. It's parasympathetic when we see it constricting, where circular muscles will constrict, and it's sympathetic when it dilates. And the reason is if you're in a fight or flight situation, we want our eyes to dilate so that we can see more, we can take in more of what's around us. All right, but guys, this happens just on its own. You don't have to tell your eyes to do this. You don't have to prepare and be like, all right, I'm about to go outside where it's bright. All right, I need my pupil to constrict, to get smaller. We don't have to think about it. It automatically happens. Okay, so this is a type of almost like a reflex, an autonomic reflex that should take place. Now, the last layer of the eye is the retina. This is called the nervous tunic. Now, the reason it's called the nervous tunic is because it contains all the nervous tissue. This lines the posterior, the back 3/4 of the eye. Now, guys, one thing to note is it doesn't come to the front part of the eye. It only covers 3/4, so it's only like a C shape. Okay, it doesn't cover the front. This is the main area where you're going to form an image and send it to the optic nerve, which you'll recall is cranial nerve number 2. Now, this has what we call pigment epithelium. It's the non-visual portion of the retina. It contains melanin similar to the choroid to help absorb light. It helps the choroid to absorb the stray light inside of the eye and be able to concentrate it towards the neural portion. The neural portion is the visual portion of the retina. In this area, there's what we call a photoreceptor layer. This photoreceptor layer contains photoreceptors that convert the light that we bring in into a potential, into ultimately an action potential that tells the optic nerve what we've been seeing and sends it to the brain. Now, there are 2 types of photoreceptors in your eye. There are rods. Rods specialize in your black and white vision. That's what the B and W means there, your black and white vision, and in your dim light. So this allows you to be able to see shapes and things like that even when there's not a lot of light present. It also allows you to see whether it's light or dark. Okay, and also movement that's around you. Okay, the other side is cones. Cones are specialized for your color vision and your vision acuity, how well you can actually see the details. The cones also help you in seeing in bright light. So both of these are super important. Okay, your rods and your cones. Also located on the retina, you're going to find some areas that are called the macula lutea. This is the exact center back of the retina. This is for your visual axis, so where the light comes in and hits at the very back is called the macula lutea. We also see that there's the central fovea. The central fovea is the area of your sharpest vision because this is going to be the area where most of your cones are located. So your rods are kind of spread out through the retina, but there's going to be a concentration of cones, which are called your centra fovea. Now, this is really important because these areas, if they get damaged, they can ultimately affect your vision, whether it's your macula due to like macular degeneration or if the central fovea gets injured. Now, from the photoreceptors, they're going to send impulses that get carried by bipolar neurons to what we call the ganglion cells. The ganglion cells then will connect at what we call the optic disc. The optic disc, guys, is called your blind spot. And the reason it's called the blind spot is it can't collect any information here. Okay, and this is where the ganglion cell axons will leave through the optic nerve. In your lab, it wants you to test your blind spot. It gives you a picture where you have an X and a circle, and it wants you to hold your book up and come in, focusing in on the X. You can see the circle towards the side in your peripheral vision. When you get to the point where you can still see the X, but the circle disappears, that's where your blind spot is located. That's where the optic nerve is actually located, and you have one on both sides. Okay, so we have those blind spots that are there. And that's part of the optic disc. So guys, in this particular picture, this one is just showing you where the optic disc is located. That's where your blind spot would be. It also shows you the retinal blood vessels, how blood vessels run through. The macula lutea is the centermost part, and then the central fovea, if you'll notice it's a little darker in color, that's where the most of your cones are going to be located for your sharpest vision. Okay, and so this, when you go to the eye doctor and they use that kind of bluish color light to look into your eye, they're actually looking at your retina here to see all these different structures to make sure that they're there and they're not being damaged. Now guys, when we look at the difference, this shows you structurally the difference between the rod and the cone. You can see where they get their name. This is one more rod shaped and the other side has the cone on the top. For your rods, guys, you have about 120 million rod photoreceptors in each eye. They're there for the low light threshold in your black and white images. A loss of these happens with age. This makes it difficult for people to drive at night. So if you've ever heard of people who have night blindness and they can't drive at night, it's normally due to them having a lower amount of rods in their eye, and this does happen with age. When you age, your vision just kind of gets worse. Okay. On the other hand, we also see the cone-shaped receptors, and you can see them again located here. The cone-shaped photoreceptors are more for your bright light, and they're to produce color images. There's 3 varieties. They're going to pick up different pigments of the color, and there's red cones, green cones, and blue cones. Now, when we look at these cones, guys, 64% of the cones are red cones. 32% are green and only 2% are blue. Okay, so there's not a real high percentage of the blue. The reason though, guys, is that the blue light actually, the blue cones have a higher sensitivity, so we don't need as many of them as we do the red light. And that's what a lot of that has to do with the wavelength of the light as it comes in. You'll see that these cones and rods collect the information. They send them over here to the neural layer, which is the bipolar cells, then to the ganglion cells, and then ultimately they'll travel to the optic nerve, which you can see here at the bottom of the picture. Now, if you have a complete loss of your cones, you are considered legally blind. Okay, and so a lot of times people, if they have a complete loss of their cones, they're considered legally blind. If you lose kind of the concentration of certain cones, like the red cones, the green cones, or the blue cones, but you have the other ones, these are going to cause different types of color blindness. And in your lab as well, there's a color blindness test that I posted that you can do to see if you have any kind of color blindness present. Alright, so now let's talk about how your eye is put into 2 different kind of cavities or chambers. And when you do the eye dissection, you're going to actually see these. There's an anterior, which is the front part of the eye, and then there's a posterior, which is the back part of the eye. Now, the lens, guys, is an avascular structure. Your lens has no blood vessels to it. It's located just behind your pupil and your iris. The whole function of your lens is to focus the light rays for clearer vision. This is to help give you visual acuity. And so people who have really good vision, they don't have to wear contacts or glasses, their lens are doing their job for them to be able to focus. Others of us, like myself, our lens is not able to project the image back on the retina like it should. It could be due to the shape being wrong or the muscles not being strong enough or something like that. And so glasses or contacts are needed to help with this. The lens also is going to be held in place by these suspendatory ligaments. So it's like a suspension bridge, kind of like the bridge in San Francisco that's held together by the suspension cables. We see that this lens divides the eyeball into its 2 cavities. It divides it into an anterior cavity, which is in front of the lens, and a posterior cavity, which is behind the lens. The posterior cavity a lot of times is known as the vitreous cavity, and you can see that here located in this picture where you have the anterior cavity and you have the vitreous chamber or posterior cavity. Now, with the anterior cavity, you're going to see that there's 2 spaces here. This is the space between the lens and the cornea. It is filled with the watery fluid that we call the aqueous humor. Remember, it is secreted by these ciliary processes behind the iris in the back chamber of this area, the posterior chamber, and the fluid has to then travel around into the anterior chamber, the front. The aqueous humor is produced by the ciliary bodies. It's traveling along the posterior surface of the iris, and it flows through the pupil, the hole, into the anterior chamber, the front of the iris. Now, this fluid does have to be drained because we want to keep the pressure in our eye constant. So when we make some of it, we need to also drain some of it. So it drains into what we call the scleral venous sinus, that little hole we talked about between the sclera and the cornea. This area is going to then drain the fluid and return it to the venous supply. It's going to return it to your blood, into the veins. Now the reason, guys, is these structures are avascular. They don't have blood vessels. So these ciliary bodies create the aqueous humor in order for them to deliver oxygen and food to these areas. But then because they're doing that, they're also going to pick up the waste products, and we need to return it back to the blood so that it can be cleaned and taken care of by the kidneys and other structures. Now, the whole point of this fluid is to help maintain an interocular pressure. We need a certain pressure in our eyeball so that it can keep its shape. If this pressure is not constant, it'll actually kind of deflate the eye. So this fluid helps keep it inflated so that it's a round structure. Now, aqueous humor, guys, gets replaced every 90 minutes. Now, let me just give you a heads up. When you do the dissection, if you open up this anterior cavity, you're going to see some fluid come out of it. That is aqueous humor. It's not going to have very much in it. You're going to actually feel that that eyeball is kind of deflated when you do the dissection. Well, the reason is because it's not constantly producing the aqueous humor because it's not connected anymore to the body to do this. All right, so that's why it kind of deflates the eye. But you'll notice that any fluid that comes out of that is going to be that aqueous humor. Now, before we move on, I want to talk to you a little bit about glaucoma. Glaucoma is normally an issue here. This is where the interocular pressure starts to increase. Okay, it increases due to either an overproduction of the aqueous humor or a blockage in the drainage of the aqueous humor. So either way, pressure is going to increase. It's going to push on the retina, and it's going to cause a lot of pain, and it's going to mess up the individual's vision. And so that's what happens with glaucoma. All right, this brings us to the vitreous chamber. The vitreous chamber is the posterior cavity or chamber. This is the space between the lens and the retina. You can see it's behind the lens. Now, this also contains a type of fluid, but it's more gel, okay? And you actually will feel this too when you dissect the eye, that it's a more gel-like structure, kind of like a jelly. It's a little more solid than the fluid from the aqueous humor, and this is called the vitreous body. The vitreous body helps maintain interocular pressure as well as the eyeball shape, and it keeps the retina smooth. Remember I told you the retina covers more in a C shape. It doesn't cover all the way around. So this actually helps hold it up against the back of the eye. If this actually starts to shrink, we'll see that the retina will actually start to fold, kind of like when wallpaper doesn't stay attached and it starts curling. Okay, because the glue isn't holding it down. The same thing can happen here if there's not enough pressure due to the vitreous body. Now, your vitreous body is formed during embryonic development, okay, in the embryonic life, and it never gets replaced, okay, through your life. Your eyes are the size that they are going to always be when you're born. This is why babies a lot of times look like they have really big eyes, but you kind of grow into them because your eyes actually don't change shape throughout your life, okay? They stay the same size. Now, as we age, shrinkage of the vitreous body can happen, and if this happens, it can cause detachment of the retina. Now, a retinal detachment is considered a medical emergency. It needs to be fixed right away. If it's not, okay, if it's not immediately repaired, it can actually cause blindness, permanent loss of vision in that area, and blindness. That's why it's very important if you have retinal detachment happen. And guys, retinal detachment a lot of times happens where you start to see a black or cloudiness that happens in part of your vision, and it will continue to grow if it is not treated. All right, and it's not really painful. A lot of times, retinal detachment has no pain with it. Okay, and so because of that, it's really important. It's still a medical emergency because a lot of times we don't go to the doctor unless there's pain involved. All right, but it is considered a medical emergency. All right, so now we've talked about the structure of the eye. Now let's talk about how the eye actually sees. All right, so how do we form images back on the retina? Well, we have to look at how light moves. We're going to look at the fact that light moves differently through air and water. So in the picture up here at the top, you can see that when light strikes water, it actually bends. Well, remember, we just talked about how your eye has aqueous humor in it, and then even in the vitreous body, it's more of a gel, so it's going to slow down the light as it goes through here. Okay, the light waves are going to slow down from air to water to the gel, okay, and allow us to be able to focus the light rays where we want. This is why when you see it's bending, like if you look down at somebody who's in a pool, it may look like their legs are big or misshapen because the light is bending. This is called refraction. Refraction is the bending of light rays. The cornea and the lens will help with this and focus the image onto your retina. Now, one thing to note is when it focuses it on the retina, the image is actually inverted, which means it's upside down, and it's reversed. It's backwards. Now, we don't see the world as upside down and backwards. Our brain actually puts it back the way it was supposed to be. It rearranges it. But when we see it on our eye, it's actually upside down and backwards in the process. Now, if refraction happens like it should, we could see it in the top of this first picture here where it's pinpointing right on the retina and we're seeing what we should. The problem is if you look in these other pictures, it's not. It's either focusing the image before it hits the retina, like you can see in B, or it's focusing it behind the retina, like you see in the picture D. Now, if we see that it's focusing in front of the retina, this is called myopia, guys, is what we call nearsightedness. This means you can see up close, you can see near, but you can't see far away, okay? So this is when a person needs to get glasses so that they can see far away and it's focused. On the other hand, we see what we call hypermetropia. This is farsightedness. This means you can see far away, but you can't see up close, okay? Because it's focusing it behind the retina. Now, this is going to be individuals who have to have reading glasses. Their arms start to, you'll see people a lot of times as they get older, they'll take a piece of paper, okay, and they're trying to read it and they'll move it until it's at that sweet spot where they can see it. Well, when their arms are no longer long enough, then they need reading glasses. We also see as people get older, they can't see far or near, and that's when they have to get bifocals. Another thing that can happen is somebody can have what we call astigmatism. Astigmatism is an irregular shaped cornea or lens. When these are not shaped properly, it causes it to be harder for them to focus the light where it needs to go. When contacts were when they first came out, people with astigmatisms could not wear contacts. They wouldn't work properly. They'd have to always wear glasses. On the other end, now they do have new contacts that are for astigmatism that help, and they can actually use the contacts as well. So guys, in the pictures over here, it shows you the different types of lenses that glasses can have that can help adjust where the focus is so that it can improve our vision due to refraction. Another thing that's really important in allowing us to see what we're looking at is called accommodation. This is focusing of the lens. For up close focusing, those ciliary muscles have to contract and the lens will actually thicken. When it thickens, it allows us to be able to focus on things up close. For distance focusing, these ciliary bodies will relax, which will flatten out the structure, and because of that, it allows it to focus when you're looking at something far away. All right, this is also why, guys, if you've ever been focusing on something up close, back here it looks blurry. Whereas when you look over at something else, what's right in front of you may look blurry. It's because of this accommodation that's taking place. One kind of disease I want to talk to you about is called presbyopia. This happens with age, and this is going to happen to anybody and everybody. It just depends on when it starts happening for you. The lens starts to lose its ability to accommodate and focus up close, and so this is why individuals will have to get bifocals or reading glasses. And it does happen to everybody. Some people, though, it happens earlier in life. Others, it's not going to happen until they're in their 70s or 80s. It just depends, but this is a natural process with age where your lens loses the ability to be able to focus the light as well. Another thing we want to look at is what we call convergence. Convergence is the inward movement of the eyeballs as they both are directed at an object being viewed. So guys, when we converge our eyes and we look at one object, it's going to allow you to see this. Now you can actually see convergence very well when you take your finger and you move it towards your nose and your eyes converge to stay on your finger. That's why they'll cross, because they're converging. The nearer the object, the greater degree of convergence that has to happen in order for you to still have binocular vision of using both eyes. Convergence helps us maintain our binocular vision and be able to see in 3 dimensions. This is why, like, if you go to a 3D movie, you need both eyes to be working. My husband has an issue where he can't see real well out of his right eye, so when we go to 3D movies, it doesn't look 3D to him. All right, he has to wear the glasses that help with 3D, but that's just so it doesn't look fuzzy. Okay, because both eyes aren't working, it doesn't look 3D in the sense of it coming at him. Also, this is going to help you with depth perception. If both eyes aren't working, it's going to be hard to tell how far away something is. Okay, so depth perception is also going to be important with convergence. The last thing here that we want to look at on this slide is constriction of the pupil, and that's what the picture down here is talking about. This is an autonomic reflex. It's just controlled by your autonomic nervous system. It has the contraction of the circular iris muscles. This allows light rays to enter only through the center of the lens. So the image is very clear and not fuzzy. So depending on how much light is present, if it's bright light, you'll see that they'll constrict and the pupil, the hole gets smaller. In normal light, it's more of a medium size, but in dim light, the pupil gets a whole lot larger. larger. Also, this is why when you go to the eye doctor and they want to dilate your pupils, they actually cause these muscles to relax, okay, and it makes them open up all the way so that they can look into your eye easier, okay, and dilate them. But it's almost like they're paralyzing these muscles though too, and that's why it takes a while for when the medicine to wear off to where you can actually do this process again. All right, so now let's talk about how we get the image to our brain and interpret it into what we're seeing. So with your visual pathway, we first have to have the light ray strike those photo pigments. So they've got to strike the rods and the cones. When this happens, those receptors, the rods and the cones, do transduction. They're going to send their signal towards the optic nerve. Okay, this releases neurotransmitters to the bipolar and horizontal cells, which then proceed to the ganglion cells through the retina straight to the optic nerve, which remember is cranial nerve number 2. Once the message enters the optic nerve, cranial nerve 2, then the optic chiasma, optic tract, okay, then it travels down the optic chiasma, which is the cross, okay, that crosses across, which you can see in the picture, and it goes through the optic tract. This tract is then going to go through the thalamus and then ultimately to the cerebral cortex where the occipital lobe is located. So guys, even though you're seeing here in the front, the very back part of your brain, your occipital lobe, is the area that's going to interpret what you're seeing. Now, some axons cross to the opposite side, which you can see in this picture, while others are going to remain uncrossed into the same side. And this depends on whether you're looking at the vision that is going to be your peripheral vision over here or your nasal vision. Okay, so when we're looking at this in this particular picture, it kind of shows you how this crossing over kind of takes place. Now this picture is found on page 593, and it just shows you how some cross over and some stay on the same side. So we have nasal vision, which is here, and then we have temporal vision here, which is your peripheral vision. So guys, this is our flow chart. We first start with light. The light travels through the cornea, which then hits the water, which is the aqueous humor. This is going to slow down the light rays. This is going to then allow it to travel through the pupil, the lens, the vitreous body. It's going to then go through to the rods and cones, who are going to communicate with the bipolar neurons, the ganglion cells, and then ultimately to optic nerve 2. Okay, cranial nerve 2. The cranial nerve then is going to allow the message to travel through the optic chiasma, the optic tract, the thalamus, and then ultimately to the cerebral cortex occipital lobe. So this is your pathway for how we get what we see out here through into our eye and be processed back by the occipital lobe. All right, and if you'll notice, this is a lot more complex than what we have seen in the past, okay, with some of the other sensations. All right, there's more steps to it than just that first neuron, second neuron, third neuron. All right, now we're going to switch and we're going to talk about the ear. The ear has 3 main regions to it, or principal regions. The first is the external ear, okay, and it's in this yellow color here. This uses air to collect and channel sound waves, so it has a specific shape so it can funnel sound into your ear. Then we have the middle ear, which is shown here in blue. This uses a bony system to amplify the sound vibrations, because sound travels in waves. So as they hit and they vibrate these bones, it allows it to amplify and make it bigger. And then the inner ear is shown here in pink. This generates action potentials to transmit sound and balance information to your brain. Okay, so you have the external ear, your middle ear, and your internal ear. So we're going to start first with the external ear. The external ear collects sound waves and passes them inward through this canal. The auricle, or what we call the pinna, which is right here, is this funnel-shaped flap of elastic cartilage and skin. Okay, and you can see it here. It's highlighted here with the auricle. Then we have the meatus. The meatus is the external auditory canal. This is a curved tube in the temporal bone, and you learned about this back when we talked about the bone in the temporal bone, the external auditory canal. This contains what we call ceruminous glands. These are sudoriferous, but they secrete cerumen, which is earwax. Okay, so they create that earwax, which is important because it helps trap dust and things like that and help keep your ears healthy. It's going to help protect the middle and inner ear. We then have the tympanic membrane. The tympanic membrane is known as your eardrum. This is a thin covering over the middle part of your ear. It's composed of simple cuboidal epithelium plus some collagen and elastic, because it is like a drum. When those waves hit it, we want it to vibrate and we want it to be able to move in and out, and that connective tissue allows it to have that structure of being able to move in and out like a drum. Now, if this drum structure gets perforated, meaning that there's a hole in it, okay, it gets a hole, and this could be due to compression of your ear, like due to diving, like you dive down too quick and come back up, it can put compression and break your eardrum. An explosion can cause the eardrum to burst. Trauma, like if somebody comes up and boxes your ears and pushes air in there real quick, it can actually do this. If there's a middle ear infection, where fluid builds up in the middle ear, it can push on the membrane and break the eardrum. There's a lot of things that can cause this, but it reduces your ability to hear because that eardrum is supposed to vibrate. It is also very painful to have a perforated eardrum. Now, the middle ear that we're going to look at is also going to be behind the tympanic cavity, or it's going to be behind the tympanic membrane, so we call it the tympanic cavity. This is an air-filled cavity in the temporal bone. It's lined with epithelium tissue. There's going to be another tube here, which is called the auditory or eustachian tube. This is made of bone and cartilage. This leads from your middle ear to your nasopharynx, so down into your throat area. It's closed at the throat end most of the time, but it does open when you yawn or you swallow, and the whole point of that is to balance the pressure in your ear and in to the outside. So this is why if you've ever had your ears pop, like when you're flying or something like that, or even when you just open your mouth wide and you go like that and your ears pop, that's that tube opening to equalize the pressure. Okay, so it equalizes pressure. Now, if the inside and the outside pressure are not balanced, your eardrum can't vibrate properly, so it can be painful. You can get a ringing in your ears, or it even can cause vertigo where the room is spinning when it shouldn't. And so it's really important for the pressure to be maintained through this tube. Now, the problem is this tube is located next to the nose in the throat. Well, if you have a cold, okay, you have an issue in your nose, or you have, like, say, strep throat in your throat area, your pharynx, it could potentially go up this tube. Okay, and so if you have a pathogen that's in your throat area or your nasal area, it can travel. If you have a pathogen in your nose or throat area, it can travel up through this tube and cause a middle ear infection, and we call those the ear infections. It's actually the middle ear, but it's an ear infection. It can cause a buildup of fluid, affect your... Now, inside this particular area of the middle ear, you're going to have what we call the auditory ossicles. These auditory ossicles are the middle ear bones. They are actual bones, but they are super tiny. And guys, I want to kind of show you here in this picture. This is the size of the bones here, and this is them up next to a dime. So they could fit on the dime very easily. So they're super small. The first one we can see here is called the malleus, or the malleus. This is the hammer-shaped bone. It's connected to the inside of the eardrum through a synovial joint. So recall back from joints. Synovial joints mean they have fluid around them. It is also articulate or joined to the incus, which is the next one. The incus is the anvil-shaped bone, and it articulates to the malleus and the stapes, which is the next one. The stapes is a stirrup-shaped bone, and it articulates with the incus as well as with this next structure we're going to talk about, which is the oval window. Now, the auditory ossicles are held in place by several ligaments and also two muscles, and you can see the ligaments here in the picture. The base of the stapes fits into an oval window. This is a thin layer of bone between the middle ear and the inner ear. It is also covered with a membrane. This membrane that's around this oval window is your secondary tympanic membrane. It's like another eardrum, it's just smaller and it's towards the back. Okay, this is why this is also known as the tympanic cavity, because there is a membrane on both sides of it. Okay, so that's what we're looking at here for the middle ear. The innermost ear, the inner ear, is composed of a labyrinth area. Okay, you can see here it has a labyrinth, which is lots of twists and tubes. There's the bony labyrinth, which is located in the perios portion of the temporal bone. So this is actually going to be a hollowed out region in the temporal bone. We see that there are semicircular canals. These are going to be important for equilibrium. You can see them here. We also see the vestibule, which is involved in equilibrium as well. There's also the structure called the cochlea. The cochlea, guys, looks like a snail shell. This contains the receptors for hearing. So when we see semicircular canals in the vestibule, that's for the equilibrium. The cochlea is for the hearing portion. The round window in the secondary tympanic membrane is near where the vestibules and cochlea meet. You can see the stapes here that's shown. Inside the cochlea is a structure called the spiral organ. You can see it creates a spiral. This is also known as the organ of Corti. It's a coiled sheet of epithelial cells, and this is your actual organ of hearing. This is what's going to communicate with the nerve and let you know what you are hearing. The organ of Corti contains thousands of hair cells. These are like those microvilli, small little extensions like we saw with the olfactory receptors, but also what we saw with the taste buds. But they're these small hairs, and as they move back and forth at different vibrations, it's going to allow you to detect different sounds. Now, inside the bony labyrinth, there's also what we call a perilymph. This is similar to cerebral spinal fluid. There's a fluid inside of here. And there's what we call the membranous labyrinth. And you can see the membranes here. It's a series of sacs and tubes inside the bony labyrinth. So they're shown here in purple and also in this kind of reddish color. And they are a lining of epithelial tissue. And they contain what we call endolymph. Endolymph is going to be inside of the membranous labyrinth. And it is similar to intracellular fluid or cytosol, the fluid that's inside your cells, like the cytoplasm. Okay, that's located there. So let's talk about hearing first before we get to balance or equilibrium. So with hearing, guys, the auricle right here is going to detect the sound wave, and it's going to send it through the external auditory canal. Okay, so and sometimes we can enhance this if we cup our ear and it catches the sound better to send it into that structure. The sound waves then strike the tympanic membrane, causing it to vibrate back and forth. So if we have a low-pitched sound, it causes slow vibrations. If it's a high-pitched sound, it causes fast vibrations. Vibrations can get conducted then through the tympanic membrane to those ossicles. So as the tympanic membrane moves back and forth, it causes the malleus to move, which causes the incus to move, which causes the stapes to move. So it sends the vibration down the middle ear. As the stapes moves, it pushes that membrane of the oval window in and out. So it's sending the message into the internal ear. The movement of the oval window causes the fluid. We talked about them being endolymph inside there in the cochlea and the perilymph. It causes that fluid to start to move. Okay, it creates waves, kind of like a wave pool. In the wave pool, they have that machine that goes up and down, which moves the water to allow it to create the waves. The same thing's happening here. That vibration is moving the fluid, so it creates waves inside the cochlea. These pressure changes due to the waves in this fluid, this perilymph, cause the round window to bulge in and out. The pressure changes in the perilymph also cause pressure changes in the endolymph. This eventually causes the hairs, those little hairs we talked about in the spiral organ, to start to being bent because fluid's going to start coming across them and they're going to bend in different ways. This is going to open mechanically gated ion channels like what we saw back in the chapter 12 for the nerves. Mechanically gated means that they get triggered by motion. So as these hairs move, they open these channels, and this creates the movement of those ions so that we can send an action potential to the nerve. Okay, it opens up the calcium channels eventually in the synaptic bulb due to the influx of the calcium ions. This triggers the exocytosis, the exit of the neurotransmitter. This neurotransmitter then talks to the nerve in the cochlear branch of the vestibular cochlear nerve, which is cranial nerve number 8. This carries the impulse to the medulla. Once it hits the medulla, it's going to then cross over to the opposite side, okay, traveling through the midbrain, the thalamus, and the cerebral cortex to the temporal lobe. So guys, what you hear on your left side here is going to cross over and be interpreted by the temporal lobe on the right side, and vice versa. So there's a crossing over because it travels through the thalamus. All right, so if you take a look here, this is showing you, when we look at this particular picture, how it's traveling. It's going to travel through the vestibular cochlear nerve to the medulla, through the rest of the brainstem. As it enters into the actual thalamus, there's some crossing over that takes place, and it talks to the primary auditory area in your cerebral cortex, which is located on the temporal lobe. Now, inside of the actual structure of the ear, this is what's happening. Now, this picture is found on page 601. So we see number 1 here, the external auditory canal sending the wave. So these little blue parts are the wave. The tympanic membrane starts to move, which then moves these particular small bones in the ear, the malleus, the incus, and the stapes. They move back and forth, which moves the fluid, which you can see here in the canal, and it causes those spiral organs to hairs to move, which sends the signal. All right, so this is just showing you kind of step by step what's happening. Like I said, in your book on page 601, it shows you a picture and it also takes you step by step. So number 1 that's labeled here in the picture, there's a little word part or blurb that tells you what's happening in number 1. Then it tells you what's happening in number 2 and so on. And again, this is found on page 601. All right, so transmission of sound to the inner ear. The route of the pathway is going to go from the outer ear to the middle ear to the inner ear. Well, doesn't that make sense? We have to come from the outside in. Remember, the outer ear is made of the pinna, the auditory canal, and the eardrum. The middle ear is the malleus, incus, stapes to the oval window. And the inner ear is going to be the vestibuli and the tympani, which is that secondary tympanic membrane. This is going to talk to the cochlear duct. This is going to stimulate the organ of Corti, which is in that cochlear region, the snail-like region, which is going to generate an impulse in the cochlear nerve. Okay, cranial nerve 8, the cochlear branch of the vestibular cochlear nerve. So here's our auditory pathway. Just like we saw in the vision side, this has a lot more steps to it. So sound is going to travel through the airwaves. It's going to vibrate the eardrum. Once the eardrum is vibrated, it's going to move through the ear ossicles, vibrating those 3 bones. They then are going to vibrate the oval window. The oval window is going to create waves in the perilymph due to its motion. This is going to move the basilar membrane. This is going to cause those small hairs in the organ of Corti to distort and bend. This then sends the signal to the cochlear branch of cranial nerve 8. Okay, this is the auditory nerve that we're talking about, okay, the cranial nerve for the hearing portion. This is going to travel through the medulla, through the thalamus, and then ultimately to the temporal lobe, where it's going to then interpret what you are hearing. All right, now we're going to switch gears real quick and talk about equilibrium and how we see equilibrium take place as well with the inner ear. Equilibrium is another function of the inner part of the ear. It is controlled by the vestibular apparatus, which is a separate section in the cochlea, and you can see it here in kind of the pinkish color. There's 2 types of equilibrium we want to talk about. There's static equilibrium that refers to the state of balance relative to your force of gravity, because gravity, guys, does affect us, right? I mean, that's that whole saying of what goes up must come down. I've never seen somebody trip and then float in the air. Okay, when they trip, they either catch themselves and keep their balance, or they fall to the ground. Okay, so the static equilibrium is helping you based on gravity. Dynamic equilibrium involves the maintenance of balance during sudden movements. Okay, so sudden movements, when you whip around to look at something, it helps you keep your balance. So there's the 2 types. Static is against gravity, dynamic is with movements. So guys, with static equilibrium, this is maintaining your body position, mainly your head, keeping your head held up relative to gravity. Now, babies, they have a hard time doing this. I mean, if you've ever seen brand new newborn babies, can they hold their head up? No, it's going all over the place, and part of that's due to gravity. The main reason that happens is because the muscles in their neck are not strong enough to hold their head up. However, once these get strong enough, we need to make sure that we hold our heads up against gravity with balance. Okay, and that's where this ureticle and saccule is going to come in. There's 2 sac-like structures inside the membranous labyrinth of the vestibule. It's an opening area called the ureticle and the saccule, and these are going to be the ones that help you with static equilibrium. They contain hair cells, okay, in here, and you can see them here in these structures, and that when we move our head, whether we tilt our head down or from side to side, they're going to move to help keep us have our balance, because when we look down like this, we don't want to fall over because of gravity just because we moved our head down. All right, so this is going to help with static equilibrium. And you can see how the fluid moves across these hairs, and it causes you to be able to adjust based on their movement. On the other hand, dynamic equilibrium is maintaining body positions, again, mainly the head, in response to sudden movements like rotation when you're going around, acceleration when you move forward real fast, or deceleration when you come to a stop. okay? So that whole idea is going to help you do this. Now, this is super important if you think about a roller coaster. Okay, there's rotation in roller coasters, whether you have corkscrews or you have loops, acceleration and going fast, and then deceleration when it comes to a stop. All this is going to be like your dynamic equilibrium. In here, you have semicircular ducts that are the primary sense organs, and if you'll notice, there are three kind of semicircular structures here. This is to help you with any way that your head moves, whether it moves up and down, like when you say yes, or it moves side to side when you say no, or when you move in a circle all together, okay? So when we look at this, these are going to help with that dynamic movement of the head. There's hair cells involved here, and you can see them in the structure, but they look a little different because they're located inside these semicircular canals. Now, the urogul and saccula also are going to be involved here, because does gravity stop just because you're moving? No. So static equilibrium has to help dynamic equilibrium because gravity still plays a role here. But dynamic equilibrium deals more with the movement part, rotation, that sort of thing of your head. Okay, and that's what we're looking at here. This is why, guys, sometimes when you get off certain rides that spin or things like that, you might feel dizzy and you might feel like your balance is off. Or if you've ever played that game where you put your head on the bat and you spin around and then you try to run and you're running like crooked, this is because you've messed up these hairs and these canals because you've spun in certain directions and it's causing them to have to reset. Eventually you'll get your balance back, but it can cause problems for a time being. It can cause problems for a short time until it resets. So let's look at the equilibrium pathway. Once generated, a nerve impulse is going to travel through the vestibular branch of cranial nerve 8, because remember it's called the vestibular cochlear nerve. So this is going to be the vestibular side. So the ascending neurons continue to the primary auditory area in the parietal lobe to provide us with conscious awareness of the position and movement of our head and our limbs. So this is going to take it to a little bit different area. It's not going to take it to the temporal lobe because we're not doing the sound part. It's actually going to go to the primary auditory area near your parietal lobe. This is to also help us be able to take in that information from those proprioceptors that are going to be talking to the somatosensory area of your parietal lobe. So this is why they're in close contact. Now, in your textbook, it talks about some homeostatic imbalances. It's really important for you to read about these. They're at the end of each chapter. There's that section about maintaining homeostasis, but also disorders. Homeostatic imbalances of the eye, there's some related here which are cataracts. That's where you get the cloudiness on the cornea. This could be due to injury, certain medications, or people who are diabetic. These can happen, but it's also common just in old age. A lot of times they'll have surgery where they'll get cataract replacements to take the old clouded cataracts off and replace them with new ones. Conjunctivitis we've already talked a little bit about, but that's the inflammation of the conjunctival membrane. A lot of times we call this pink eye. It could be a viral infection, bacterial infection, or allergies. We also see age-related macular degeneration. Remember in the eye we talked about the macula area being the middle area, which allows for the middle part of your vision to come in. Macular degeneration is what can happen after 50, and this does happen with age. And guys, the picture down here shows you the macula is the middle area. So if you're looking straight at, these are two kids, this is a kid holding the ball. What's missing? Their face, because what do you normally focus on when you look at somebody? Well, you should focus on their face. All right, so you should be looking at their face, and that's why you don't see the face here, because it's missing because it's the middle portion. If you want to see the face, they would have to look at from their peripheral vision, which still is not going to give them a really clear picture. Okay, so that's macular degeneration. For the ear, there's a number of things. There's myringitis. This is an inflammation of the eardrum. Okay. We also see infections of the middle ear. This is odious media. Odious means ear. Media means middle ear. We see this is very common in children between the ages of 6 months and 5 years old. This makes the child very irritable. They may cry a lot. When you look into their ear at the tympanic membrane, it might be very angry and red looking and bulging, especially if there's fluid in the middle ear. We also see odious externia, and that's what you actually see here in the picture. This is external issue of the ear or infection of the ear. A lot of times it's called swimmer's ear. It could be infectious, like you get an infection like bacterial or viral, but it could also be non-infectious due to an irritation of like chlorine or water getting in your ears. And this is why a lot of swimmers a lot of times will wear earplugs if they swim a lot. But also it's really important not to share earplugs, because that's when you can actually get the infection form, because you're sharing things that went into a cavity. So this is an inflamed tissue of the outer ear and the ear canal. There's two types of deafness when we talk about hearing. There's conduction deafness. This is when something hampers sound conduction to the fluids of the inner ear. This could be where you have too much earwax in your ear and it's impacted, and so it makes your hearing muffled. We could remove that earwax. One thing though, guys, with removing earwax, do not put anything smaller than your finger, your little finger in your ear. You'll notice it doesn't go very far. This includes a Q-tip. Do not use a Q-tip to clean out your ears, because it can compact the earwax closer to your eardrum and cause problems and potential infections. Okay, you should just use a wet rag and kind of do it. You can also use alcohol. Okay, if you drop a few drops of alcohol in your ear, it can help kind of clean that out with the earwax and that sort of thing. But if it gets impacted too much, you may need to see a doctor to get that extra earwax pulled out. Perforated eardrum, if the eardrum has a hole, it can cause conduction deafness. Osteosclerosis, this is where those ossicles, those tiny bones, they can't vibrate. It's almost like a type of arthritis in the inner ear. This happens with age, and so when it causes them to not be able to vibrate, it also hinders the hearing. We also see there's sensorineural deafness. This results from damage to the neural structures like the cochlear hairs inside the cochlea, or it could even be the nerve itself. All right, conduction deafness, a lot of times if the issue gets fixed or repaired, hearing can be improved, and hearing aids can help with this. But the sensorineural deafness, a lot of times they can't be improved, especially if it's the nerve that's the problem. If it's the cochlea, they could potentially have cochlea implants, which can help with hearing. But if the nerve itself is damaged, then hearing may be lost completely. The last thing that's talked about here is Meniere's disease. Meniere's disease is a disorder of the inner ear. It can affect the hearing, but one thing it also affects is balance. This is thought to be due to an increased pressure within that middle ear where the endolymph gets made in excessive amounts. This causes the patient to have episodes of vertigo, which they feel like the room's spinning even though they haven't done like a roller coaster, or they haven't spun around or anything like that. It makes it where it's spinning when it shouldn't. They may also have a ringing in their ear that we call tinnitus, where they have that ringing. It could be a mild annoyance where it's just kind of bugs them a little bit, but it could also be chronic and disabling, where they cannot continue to work and things like that because the room's spinning so much it makes them sick to their stomach. It could cause them to lose their balance. The ringing in their ears could be so great that they can't concentrate on anything else, and so Meniere's disease can be a big problem. There's not a lot to help somebody with Meniere's disease. There's not a lot of research out there. It's one of those things that not very many people have it, and so research is kind of limited. But they do think that a low sodium diet helps, because the low sodium helps you not retain water as much, and the endolymph is very similar to water, and it could collect in the middle ear. So it's important to maybe watch your diet and that sort of thing. But that's Meniere's disease. All right, so that's all that's covered here with your special senses. If you have any questions or concerns, please feel free to contact me.